Let's talk about the myofilaments of skeletal muscle in some more detail. Some quick review of what we discussed in the last video. In purple or lavender here, we see the thick filament, which are composed of myosin molecules. These are the myosin heads. In green are the thin filaments. Each thin filament is composed of G-actin molecules. So each one of these green circles is a G-actin molecule. And a thin filament is composed of two strands of G-actin molecules. So here is one strand of a G-actin molecule, and here is the other strand of the G-actin molecules. A strand of G-actin molecules is referred to as F-actin. So G-actin is the monomeric subunit. F-actin is the polymer composed of all of those G-actin subunits. We also have two regulatory proteins seen in this picture. One is tropomyosin, which we see in blue. The other is troponin, which we see in orange here. Tropomyosin in this conformation is blocking the binding site. That is to say, it's preventing the myosin head of the thick filament from binding or grabbing the thin filament. So in this region of the G-actin molecules, right about here, there is the binding site that the myosin head would want to bind to, somewhat like a magnet. It can't in this orientation because tropomyosin is blocking that binding site. To be sure, there is also a binding site for the myosin heads on each of these G-actin molecules. The way I have it drawn two-dimensionally, it's hard for me to represent that. And the same holds true down here. There would be binding sites on all of these G-actin molecules for the myosin head. But once again, the myosin head cannot gain access to those binding sites the way we see it here, because tropomyosin is blocking the binding site. The big difference in this image is tropomyosin is now moved up and out of the way, liberating the binding sites on each G-actin molecule, which would be right in this general area. And it would also liberate the binding sites on this top row, but once again, just the way I've drawn it, it doesn't really look that way, but please understand it is liberating all the G-actin binding sites. Now, what causes this is the binding of calcium to troponin. So if we move back to this previous slide right here, there is no calcium bind to troponin. Troponin is the binding site for calcium. When calcium binds to troponin, it causes what's known as a conformational change in this protein. Troponin is a protein. And the conformational change, or the change of shape of troponin, causes it to pull tropomyosin up and out of the way of the binding site, liberating the binding site, allowing the myosin head to grab that binding site. So we need calcium binding to troponin to liberate the myosin head binding site. Put another way, we need calcium binding to troponin to move tropomyosin up and out of the way. Please think about where is that calcium coming from? And then eventually, we're going to get muscle contraction or sliding of the filaments. One thing I want to point out here is the space between these two adjacent thin filaments from here to here is known as the H zone. That applies down here as well. This is the H zone right here. During muscle contraction, the thick filament is not moving, but the myosin heads are grabbing these adjacent thin filaments and moving them closer to each other. So what we see now, after the myosin heads have fired, the H zone has diminished dramatically, and that is muscle contraction. Once again, this cannot occur in the absence of calcium. Here in this picture, this is just showing the myosin head binding sites. In orange, this long structure right here is tropomyosin. This right here is troponin. So troponin is found along the length of the thin filament, not really how I've drawn it in the previous images. And what we see here, tropomyosin is blocking the binding site. It is blocking the binding site because there is no calcium present. If we look at this image, we see calcium is now present. This represents calcium. It's bound to troponin. And now we see tropomyosin has liberated the binding site. It is no longer blocking that binding site on either of these F-actin strands. As a result, the myosin head can grab that binding site. In this image, what I want to show you right here in blue is the thick filaments composed of myosin molecules, and in red are the adjacent thin filaments composed of G-actin and F-actin molecules. The space between the adjacent thin filaments is known as the H-zone. Notice the width of the H-zone. During muscle contraction, the H-zone diminishes from this length to this length right here, and we can see that now these thin filaments are much closer together. That is muscle contraction.